Hello friends, let's talk about loop diuretics today. And before going into details about the loop diuretics, I will like to explain the the mechanisms which are operative in the loop of Henle, and especially the thick ascending loop of Henle. As I have told you in a previous video that uh, the loop of Henle, it has got three parts. One is a thin descending segment which goes down into the medulla, then it takes a U-turn, and then it returns back into the cortex, first as the thin ascending limb of loop of Henle, and then it converts into the thick ascending limb of loop of Henle. So important thing to tell you here is that the descending loop is freely permeable to water, while the ascending thin as well as thick both are practically impermeable to water. So uh, that means there will be no uh, loss of water from these to the exteriors of the lumen. So now another very important thing is this segment is responsible for handling of or you can say reabsorption of around 25% of the total tubular load of sodium. So it becomes a very important site. I have also discussed about carbonic anhydrous inhibitors, and uh, they act at a very proximal site, and that site actually handles 65% of sodium. But those diuretics are not very effective. The reason being, uh, after that comes this loop, and this loop has a large reabsorptive capacity which can reabsorb whatever has been left out from that site whenever carbonic anhydrous inhibitors are given. So this site becomes very important for diuretic action, and the diuretics which act here are called as loop diuretics. So I'll tell you about that, but before that I'll be telling these mechanisms which are present here. So as you can see, this is the luminal side, this is the cells which are lining this lumen, and then there is the medullary interstitium. Since this part, it lies in the medulla, and this is a thick ascending limb of loop of Henle. So the one very important channel, or you can say symport, is present here, and this is called as, it has been named as NKCC2. This channel and this symport, you can say, and it leads to movement of one sodium, one potassium, and two chloride inside the tubular cell. And when this happens, you can see there are two positive charges and two negative charges. So this movement is electronically neutral. There is no net gain of charge, and this is what happens. And in addition to that, in every cell, there is one sodium potassium ATPase. This is sodium potassium ATPase, which leads to uphill transport of sodium out of the cell, and downhill and uphill movement of potassium into the cell, and this is energy consuming. So this is one channel which you need to know about in most of the cases, everywhere, I would say, not only in reference to renal cells, but everywhere this pump is active, and this is responsible for, this is what is responsible for movement of this channel, because it decreases the sodium inside the cell. So sodium goes into and also takes along potassium and two chlorides. So now what happens inside this, there will be sodium, so sodium will be pumped out from this. Chloride and potassium, they also go out, but potassium is entering by two ways, also from the lumen, and another way is from the medullary interstitium. So there is excess of potassium inside the tubular cell, and there are some leak channels present which open into the luminal side, which allow potassium to move out into the lumen again. So when potassium moves out from this cell in direction of its concentration gradient, obviously it becomes more concentrated inside the this tubular cell, so it has a tendency to move out, and there are channels which help it to move out. So when potassium moves out, what will happen? Now this region will become positive because a positive ion is moving out of the tubular cell. When this becomes positive, there will be a push for a positively charged ions to move away from the tubular lumen. So what happens is, whenever this potassium moves out, the divalent cations like calcium and magnesium, they are carried by means of a paracellular pathway again into the interstitium. So this is how this movement of potassium out of the tubular cell into the lumen helps in the reabsorption of calcium and magnesium. So this is what normally happens, and this thing happens in the medullary interstitium. So whatever these ions you have taken away from the lumen, they are going into the medullary interstitium, and medullary interstitium concentrates these ions, and this movement of these ions is actually responsible for what we call as a medullary hyperosmolarity. And this medullary hyperosmolarity is very important for the final concentration of urine, which I will be discussing later, but I will be giving an account of that also here. So medullary interstitium, whenever so many ions they go and they get collected here, they are not reabsorbed properly from the interstitium due to a countercurrent system provided by the vasa recta, which supplies this medullary interstitium. So the ions are kind of trapped inside this medullary interstitium, and what they do is they provide a gradient, an osmotic gradient for movement of substances, especially water, that I'll be discussing. Now what happens is, the loop diuretics, they act at this site, and whenever they act at this site, what will happen? This symport is blocked. When this symport is blocked, what will be the consequences? One thing is, when sodium does not enter, potassium does not enter, chloride does not enter, there will be no net movement of ions into the medullary interstitium. So the medullary hyperosmolarity will be dissipated. So the hyperosmolarity which was responsible for concentrating urine, it will be lost. So in a way, the concentrating ability of the kidneys, it will be reduced. So that is one thing. Another thing is, since no potassium is going inside, no potassium will come out. So this process is also blocked, and when this process is blocked, obviously there will be no reabsorption of calcium and magnesium, and they will find a way in the urine. So they can lead to what? They will find a way in the urine. So that will lead to what? A net loss of calcium and a net loss of magnesium. So this is two important things which will happen whenever you are giving a loop diuretic. And since I have told you that this segment leads to handling of say 25% of the total the sodium load, so there will be a lot of sodium which will also be entering into the urine. A lot of sodium will be presented to the further distal segments, and the further segments, they do not have the capability to reabsorb much of the sodium. That's why these diuretics, they are called as what we call as high efficacy or high ceiling diuretics. They are very, very effective in causing diuresis. Okay. Now again, you know, one more thing, since I have told you that this thick ascending limb of loop of Henle is impermeable to water. So normally when there were no diuretics, this would lead to dilution of the tubular fluid, because the ions would be moving but there was no net movement of water, so there would be a lot of water, excess of water in the lumen, and these ions only will be reabsorbed. So this will be diluted, and that's why we also call this as the diluting segment. Now when you have given a diuretic, what will happen? These ions will not go into the tubular cells, they will also stay here. So the dilution of the urine will also be suppressed. So giving a loop diuretic will hamper both the diluting capacity as well as the concentrating capacity of the kidneys. And this can be better understood in terms of what we call as free water clearance. So free water clearance is a term which is given to understand how much extra water is being excreted whenever the urine is passed isosmotically. Means as many electrolytes you have to lose, like waste materials you have to lose, and if they are lost isosmotically, that means having the same osmolarity as that of the plasma, whatever additional water is presented along with that, that is called as a free water clearance. So whenever it is more than whenever the water content is more than what is required to excrete those waste products isosmotically, that would be called as a positive free clearance, and that will happen whenever the urine is dilute. Okay, whenever the urine is dilute, obviously there will be extra water coming, so that would be called as a positive free water clearance. And whenever a concentrated urine is formed, means you are reducing the water content, and that is called as a negative free water clearance. So now, since these loop diuretics, they interfere with diluting capacity as well as concentrating capacity, so what will happen is they will reduce both the positive as well as the negative free water clearance. Is that okay? So both these things they will be interfered. Now another thing is, since a lot of sodium is now presented to the distal tubule, what will happen? The distal tubule, there are some principal cells present in the distal tubule where the sodium is reabsorbed and potassium as well as hydrogen is exchanged at that point. So a lot of sodium is reabsorbed at that site, but in exchange for potassium or hydrogen ions. So that will lead to what? That can lead to, again, since a lot of sodium is presented, a lot of potassium will be coming out, a lot of hydrogen will be coming out. So two things can happen. One is hypokalemia, because the potassium content will be lost, so leading to decrease in potassium, what we call as hypokalemia. And another thing is, since hydrogen ions are also lost, so hydrogen is, it is acidic. Whenever hydrogen is lost, another thing that can happen is alkalosis. So these two things which you can call, they are very important adverse effects which you can see with this agent, these loop diuretics. And another is, since magnesium will not be reabsorbed, so there will be loss of magnesium also. So hypomagnesemia is another adverse effect which you can see. Hypocalcemia can also happen, but normally it does not happen because other mechanisms are available which restore the calcium levels. So these are some important adverse effects which you can see with the loop diuretics. In addition to that, there is one more. These loop diuretics, they are also ototoxic, means they are toxic to the airs. They can interfere with hearing. So that is another important adverse effect with them. Then they also, since whenever they cause a brisk diuresis, they also reduce the total body volume. So the body volume of water, or what you can say is hypovolemia is caused, and whenever there is hypovolemia, obviously the reabsorption mechanisms, they become more aggressive. So they lead to increased uric acid reabsorption in the proximal tubule, and whenever uric acid is more reabsorbed, what will happen? There will be chances of hyperuricemia. So that is also one important adverse effect which you can see with these agents. The agents which are classified under this loop diuretics, they are furosemide, bumetanide, torsemide and ithacrenic acid. These are four agents commonly used. And then there is one very important mechanism which through which these loop diuretics also act, and that is by increasing the synthesis of what we call as prostaglandins, because they induce the formation of an enzyme what we call as cyclooxygenase 2. So this cyclooxygenase 2 leads to formation of prostaglandins, and these prostaglandins, they are vasodilators. So leading to vasodilatation, they increase the capacitance of the veins. So whenever the veins are dilated, they can accommodate more blood. So the edematous states like pulmonary edema or edema anywhere in the body, it can be these loop diuretics, they become beneficial in those conditions also. So that was about the loop diuretics. I will be continuing with this series of lectures on diuretics. Thank you so much.